מיקי, עדי, בישרה עווד, חסר לי, אוקיי. הרן שני, הרן שני, אז יש לנו עוד דקה. דניאל הרשקוביץ, מתן, לאה, אחלה, אורי, אורי אני חושב, נכון? לא. ומינה? מינה, את לא רשומה לקורס משום מה. כן מה?
כן, בעיות מזכרות שביקשתי לקרוא לבד.
אוקיי, אז רשום עם פנינה ואני לא ידעתי מה שמך שם משפחה ולא ידעתי איך לשבץ אותה, אז זה מבוסס על סיפור אמיתי. פנינה, נכון? ומי? זו שונית שהיא לא רשומה לקורס, ואת?
I'm also not signed up yet.
You're not signed up yet?
But, yeah, there's a problem with the enrollment. So I should be signed up really soon.
No, you should tell me because I'm, you know, it's money time now. We have to set the schedule of the. What's your name?
S-H-A-N-A, Shayna.
Shayna? Okay. And you are?
Haran.
Haran Shani?
כן.
Okay. ובישרה חסר לי. Okay. Okay, so everyone is here. Great. Okay, so now I know. You guys have to enroll into, make sure that you're signed up. Okay, and we'll see how we do that. I think we have 2 slots here. Okay. So given the... fine, so that's it for the Volga. Just some administrative issues, given the... You hear me? Good? Okay. So given the final number now that I know, I hope I know the final number of participants, I will play around with the seminars. I will still change your dates. Besides the first 3, I will still swap the dates because we want to make sure everybody presents once and that we have at least a few tutorials like the ones we're going to try out today. The paper you should have read for class. Okay? Okay. So today we're going to do 2 things. In the first hour, I'm going to give you a demo on what am I expecting from you to present. This is generally true for your academic life, your scientific life, when you are, and you will start soon enough to be part of a lab, present data, etc., etc. So this is going to be semi-tutorial, semi-scientific. The scientific part is of less importance in the first hour. And the paper that I chose to do is one to ease my work, is one that I'm involved in. And the second part, we're going to talk about the classical Hubel and Wiesel paper. So I have another person in class.
Hi.
Name?
Nimrod.
You're signed in?
Free listener.
Free listener, I see. Okay, for free, no problem. Welcome. Okay, so today we're going to talk in the first hour about a paper from Neuron in 2011 entitled Multisensory Integration of Natural Odors and Sounds in the Auditory Cortex. The authors are Lior Cohen, Gidon Rothschild, and myself. And whenever you present a paper, first of all, you have to put it up, the title up, so everybody knows the title of the paper, who wrote it, etc., and of course to give credit to the people who did that. If you don't know, normally people in the natural sciences, this can be different in other sciences, the people who did the work by importance are the first ones. So the first one listed is the person who actually did the work, and he's listed first. So if you're working in a lab in biology, for example, and you did the work, you should be first on the paper. And normally the senior author is the last author. These days there are all kinds of combinations with asterisks, people who contributed similarly, etc. But basically you should first, when you talk about the paper, you should acknowledge the people who did the work. And of course, the journal. And I already see I omitted that this paper was published in 2011. Okay, then you have to give a general background on the topic of the paper. Notice that in the slides that I'm using, and of course, if you have any questions, stop me at any time. You should use the space that is given by the white background here. Don't work on, very small bits and pieces of the slides. Use the space that you have. In this case, I put up 3 pictures that would convey what I want to say. And perhaps one of the most important things when you give a talk is to keep things simple. Less is more. Okay? If you understand that, you are halfway through on giving a good presentation. So less is more. Don't pack your slides with tons of information. Don't put anything that you are not going to use. Everything that is on the board must be used. People in the audience will read and look at what you put on the board. If you want to convey some message and you don't want people to look at the numbers and graphs, you press the B key on the keyboard, and this makes this disappear. Once you do that, this is a magical trick. People immediately put your attention to you. And in many cases, what you have to say is extremely important. And if you want people to listen to you, this is a very good trick to get them off the board. Okay? So today, or this paper, has to do with motherhood. Motherhood is becoming a mother. And this is a very basic phenomena across the animal kingdom. Okay, what I wanted to convey in the first slide is that you can see this across the animal kingdom, and it also attaches people to the talk, some cute animals, and people can understand, or even just by looking at these images, see that the mothers are, their primary role here is to take care of the babies. And the essence, or the topic, the theme of this paper is about mothers, about brain plasticity, what happens in the brain of an animal when it becomes a mother. And this is not just a small, non-important detail in brain plasticity and brain in general, but rather a very basic theme that has impact across the animal kingdom, and you can find it all over the place. Not always. You can, add anecdotes, etc. For example, the rabbit is a well-known bad mother. If you read the story of Dira Laskir, you know that, if you're an Israeli boy. But the rabbits normally spend about less than 1% of the time with their pups. And all the time they are in the outside of the nest, and they're feeding, etc. And they come for a few minutes, and then they leave, just so they get the milk instantly. This has some consequences about the pups, but I will talk about this when we talk about olfaction. Yeah, shoot.
I have a general question about presentation.
Yeah.
What level of awareness do you think your audience has of your paper or the field in general?
So you have to adjust it to the particular audience that you have in class. I adjusted this talk to you. On Saturday, I gave a talk in the Cinematheque where the general audience came in, and I gave a similar topic, but I cut 90% of the statistics, and I was just talking, generally. So it depends. You're going to a meeting which is an auditory meeting. It's hardcore details. The introduction is very short, etc. We are assuming that the students here read the papers in class and have the knowledge which is of first-year grad students in the best PhD program in the Hebrew University. In Israel, the best neuroscience program in Israel. I'm recorded now. Wow. Very bad thing. Okay. So basically, you have to keep it simple. There is a title. Becoming a mother is a significant event across the animal kingdom. But as biologists, and if we want to go deeper into the mechanisms, we have to choose an animal model. The animal model that was chosen by these authors here is the mouse, the laboratory mouse. The mouse is a powerful animal model because of genetics, etc., which does not come handy in this particular paper. But the general is a very common and powerful. Rodents are very powerful animal models in biology as a whole. And what this slide wants to convey is that motherhood is also part of the rodent's life. As you can see here, mothers are normally spend about 21 days pregnant, and then they will give birth to their offsprings between 5 to 12, 13, depending on the strain. And once this happens, there's a switch, and this becomes a new animal. Behaviorally, it's very powerful to see the changes that these animals undergo. And the behavioral changes come in a variety of forms. You can see them start licking the pups, changing in the nursing position, building nests, retrieving the pups back home, etc., etc. And all of these have real-life consequences. You can really see these behaviors in the field, if you will. Okay? I'm still in the introductory part, and I'm already delving now into the paper. Okay? So what I do here is I want to give two video shots of two mothers. Now, these changes in the mothers occur because they were not there when the animal was a virgin. And the authors here in this paper just want to demonstrate how powerful one of the behaviors is. And they used an assay called, a bioassay called the pup retrieval behavior. On the left-hand side on the bottom, there is a mock nest. And now you want to get your audience interested, if you will. So you give a small game. You don't always have to do that, but there is a game here. And now the audience have to choose which one is the mother and which one is the virgin. And I play these video streams. And there's an experiment here. Basically, the pups were stolen from the mother, and they are retrieved into they're put back into the cage. And the movie, of course, is sped up. And what you can see, one animal is a mother, one is a virgin, and the behavior is very, very clear. Okay, on the left-hand side on the bottom is the mock nest, which the animals get used to the nest. And as you can see very clearly, the mother is the animal on the left-hand side. And when she finds the pup outside the nest, she will retrieve him back home very rapidly. And the control, which is in fact her sister, her litter mate, which was never pregnant, never saw a male, will, of course, relate to this new stimulus, but she never saw these stimuli, and will not retrieve the pups back to the cage. And there's also basic statistics in the paper to see how powerful this behavior is. All the mothers brought all the 5 pups. Naive virgins never brought any pup back to the cage. Yes?
All the pups are not her own, or some of them are her own?
Those are her own pups, of course. Yeah. In this assay, they are her own pups, yeah.
Do you see this behavior otherwise? Also,
I'll talk about that later on. In terms of the science, yeah, sure, shoot.
Will the same occur if the virgin one was exposed to pup?
We'll talk about that later, okay? It's very clear you did not read the paper. So, okay. So basically, but I want to talk about the outline of the talk, okay? This is a demo on how the talk should be. You see, it's a very simple slide. There's nothing here which I'm not using. There's a title, pup retrieval behavior in the lab. On the left side is the mother. P3 is postnatal day 3, 3 days after she gave birth. And this is the statistics. Very easy. Of course, at some level, especially in your papers, you're going to read complicated papers, which are mathematically complex, statistically complex. And you're going to have to make decisions on what to cut down and what to leave in the paper. And this selection of what is important and what is less important is in your hands, and this is where you're actually being. Okay, the topic of this paper is about multisensory integrations, on how the animals integrate different senses to find the pup or changes in sensory coding in the brain. And in fact, this is not a new idea. In 1974, Smotherman, Bell, Stalzek, and Elias did a behavioral experiment where they challenged the mothers with the olfactory cues and auditory cues. And they suggest in their very simple bioassay of pup retrieval, their data indicates that olfactory and ultrasonic cues from the pups interact to produce successful retrieval behavior. What I didn't tell you in the first slide, or maybe it's, is that the mothers identifies the pups based on odor cues and based on ultrasonic auditory cues. When the pups get cold, they will start to call their mother, and the mother she uses one of these cues to find the babies. Okay, and the paper here deals with how these two senses interact in a novel way in the mothers. But this is not a novel idea in the fact that already in 1974, a bioassay showed this or suggested that odors and sounds interact.
You say the same difference will occur even the right hand with the mother, but not of that this group.
No, I didn't say that yet. Okay, I didn't say that yet, and this was not tested in this paper. Okay, I'm presenting you this paper, what has been done in this paper. Okay, so far we're going to talk about motherhood. You know that motherhood is also something we can spot in mice, and we already have a bioassay that can differentiate between mothers and non-mothers. So first of all, what I wanted to say also, if you want, sometimes, some people write down when they want to give a talk. I normally put on the board points that will remind me of what I want to say. So for example, there's a title there, pup smells are salient to maternal behaviors, which is in fact what I just told you, that these two senses are important to the mothers. And you can put it up on the board. That will not interfere too much with the students, but will help you to remember what you want to say, so what you say is coherent and makes sense. Okay, so the paper actually deals with two questions. How do the mothers encode the natural stimuli of the odors and the sounds, and whether these two interact, and if so, in what way? The authors of this paper decided to study the auditory cortex. The auditory cortex is shown here on this slide with the red circle, and you can see that it is not the first or second station in the brain, but rather several stations up from the cochlea through the lower parts of the brainstem. As you know, most senses go through the thalamus and then to the cortex, reach the cortex. The cortex has been studied because the cortex is known, the neocortex is known to be implicated in plasticity as a general rule. Okay, so the first thing they decided to do is to see whether the changes in mothers could be seen already at the level of the primary auditory cortex. For some people, this would be, if you work in the cochlea, it would be higher level, but normally this is considered a very basic one of the first sensory processing station of auditory information. So now, again, you see the slide is very simple. You don't have to go all the details, but just so people know where these authors put their electrodes. Primary auditory cortex.
Where did you get that picture? Where did you find that?
So this, for example, is from a textbook, I think, or on the web. Okay. Now, again, I tried not to put many pictures here which are not part of the paper itself, but the pictures of the mothers, etc., and that were taken from the web, which is fair enough. Okay. Okay. So did they test this question? This paper is from tip to toe electrophysiological, and they used a recording technique called loose patch recording, which is basically lowering your electrode until the resistance of the tip of the electrode increases, so you know you hit a neuron. And then once they have a neuron under their electrode, a single neuron, they start recording. What they did here is record from the left auditory cortex and played sound to the right ear. You know, or if you don't know, you should know that most senses cross, and you can record dominantly, if you want predominantly to get responses from the right ear, you will find them in the left part of the cortex, and vice versa. So the basic design was to play sounds to this animal to see how does the neurons in the auditory cortex encode sounds under two different conditions. One is an air condition, where they just stream air to the nose of the animal. And the second is a condition where they put pups into a small jar. They heat up the jar, so these pups are happy and quiet. They're cuddled together like a ball. And then they stream the air through the room of the pups, so the mother can smell the pups, and then test two questions. First of all, how do mothers versus the naive virgins process sensory information? And the second of all is whether these responses in the auditory cortex change when you expose the animal to the odor of our pups. The first demonstration in figure 1b shows that if you just record spontaneously from a neuron in the cortex, this is now 20 minutes of recording. It's a very long time of recording. You see that there is some firing pattern. They fire at, in this case, at a moderate spontaneous firing rate, about 1.5 hertz. Okay? And when you open the odor valve, something happens. It takes about, about a minute or so to kick in. But in this neuron, for example, there is a decrease in firing rate, and this effect slowly recovers. It takes a few. So this is some kind of neuromodulatory interaction between these two senses. You see that when you open, when the mother smells the odor, something changes in the neurons in the auditory cortex. So this is a first indication that they're onto something here. Yes?
All the time you play sound for the...
Yes, I'll show. So basically what you do is you record and you play sound and get receptive fields of the neurons, how they respond.
It's not really spontaneous activity.
No, this example is a spontaneous activity. Okay? This is not the real experiment yet. This is just an example that something happens, and it's going to be a little bit difficult to see it later because you're going to see, before and after odor, etc. This is how 15 minutes of recordings look like, just spontaneously. Sometimes you want to give details of the experiments, not always, but you can go through this. Again, these details were not here when I talked about the prep. Now I can put the details up on the board, and they are not interfering with what you already know. If they were here in advance, you would start reading what's up here. So this brings me to the point of using wisely the animation options in PowerPoint. Okay, this is a very slippery road for many students which like fireworks very much, and they are very good at animation. So, you walk into class and things flip over and make noises and in extremely different colors. It's going to work against you if you're going to use animation in a bad way. But something like that, where you want to convey a message, first and then do something else, is a very good way of using animation because it makes the things coherent. Okay, so in this work, they used cell-attached recording because there are different methods of recording. This can impact the results. They used anesthetized mice, ketamine domito anesthesia. They used a certain mouse strain called NMRI, which is a white mouse, which is particularly good in auditory behavioral tests. And they use two different stimulus protocols. The first one is called the pure tone protocol. Used 15 different tones and some natural stimuli. And this is the details of the stimulus, which is not critical to understand the paper, so we can maybe skip it. Okay, so how does the data look like? This, what you see here on the board, are 3 neurons, 2 of which are from a mother, the one on the top and the one on the left here, and 1 is from a virgin mouse. The first panel, and each block shows 2 things. One, on the x-axis is time, from 0 to 500 milliseconds, so about half a second. And on the y-axis is frequency. Here we tested from 1 kilohertz to 40 kilohertz. And basically what one can do is get the receptor field profile of the cell. In this case, it's just the frequency over time, so you can see when the neurons fired and to what frequencies. So for example, this would be a neuron responding to frequencies from 12 to 36 or so here, and responding very abruptly to the stimulus, which is given between time 0 and 50 milliseconds. Okay? This graph shows 2 things. One is that the effects of there are effects of odors on the auditory responses, and these are only lactating mothers, and these are also reversible. So let's start with the first one. This is the recording of a neuron from a mother. So you record under air condition, air 1, and you see this neuron is pretty quiet. It doesn't spike very much, here and there. This is an example of the 20 trials for the 11 kilohertz. And then what they do is it takes about 20, 30 minutes to get the response profile of this cell, which is quite quiet. And then they repeat another time this recording. And you can see that nothing much has changed. Okay, air 1, air 2 is just the second episode of air. And then what happens is that they open the flask to the odor. So now the mother smells the odors of her pups, and they record again. It's the same neuron again. And now suddenly something happens. In this case, the neuron now responds very strongly to 11 kilohertz. You can see the rasters here, here, and here for this particular frequency. And then they close the shutter of the mouse pups, give it a few minutes to relax, and they record the neuron again. So basically, like in pharmacology experiments, you record, and the statistical tests that are considered in the test are ANOVA tests, which examine the statistical significance of the different groups in the experiment. Basically, they compare air 1 to air 2, and air 1 to air 3, air 2 to air 3, et cetera, et cetera. These should not be significant. And then air 1 to pup odors is significant, air 2 to pup odor is significant, and air 3 to pup odor is significant. Only then they conclude, indeed, that the experimental conditions are stable. See, neurons are stable. There is a change, and this change is reversible. Okay, so some of the statistical tests in the paper, if you want to go into them, you can. If you don't want to go into them, you don't have to. It depends on what you decide. But basically, this is an examination of the time of the experiment. How does an experiment look like? It takes about 2 hours to record one of these cells. And a good student, or a good postdoc in this case, comes in the morning at 8 o'clock, gets 5, 6 cells from an animal, and that's the yield of the experiment. This is another neuron showing another example. Now also an intense amplification of the response by the pup odor. And the issue of this is to show that this thing happens in lactating mothers and not in naive virgins. This is an example from a virgin animal where you can get the responses to the tone. And basically, there's no significant difference between and after pup odors. And throughout the paper shows 2 panels, 1 and 2. And this is basically a combination of before and after, okay, not the wash. It's always, so in many cases, these two panels are averaged into one, and you see this on the left side, and there's before and after, so you can see the difference. This shows you or gives you an example of how raw data looks like in such an experiment, okay? Great. So we conveyed three messages. One, we discovered that pop odors modulate tone-evoked responses in A1. This is reversible, and this happens in lactating mothers.
Actually, the difference is in the initial condition.
What? No. In this, okay, in this example, what the authors chose to show neurons that responded to the same tone, in this case it was 11 kilohertz. Every time you record a neuron, sometimes you find a neuron responding to 3 kilohertz, sometimes it's an off response, sometimes it's a sustained response, etc., etc. In this case, we chose a neuron that was significantly responsive to 11 kilohertz in all the animals just to show a given frequency. It's almost impossible to find the same neurons in different animals with exactly the same conditions, okay, to show the responses. Some have very narrow receptive fields, some have wide receptive fields, etc. Okay, but to answer your question, the answer is no. And this brings me to the next slide. So your question is good transformation to the next slide. Okay, now what you saw here are three examples, 3 neurons. Now let's see all the neurons that were recorded. What you see here is the spontaneous activity and the sound evoked activity of all the neurons that were recorded in the two different groups. In the naive virgin groups is shown on the top panels, and the lactating mothers is shown on the bottom panels. What is actually plotted here is the absolute real value of the in hertz of the spiking activity in the spontaneous activity and in the evoked. You see that spontaneous activity, this is log scale, are of course much lower than the evoked responses. And two conditions are plotted: the response of a given neuron in air condition and the response of the same neuron in odor condition. So basically, if a neuron did not change its responsiveness, regardless of what was the basal activity, he will fall on the diagonal. Okay? So what you can see is that neurons in virgins, before and after they were presented with pop odors, fall on the diagonal. And now we're seeing all the spectrum of the odors. Some neurons fire at 0.05 hertz. Some neurons fire at 5 hertz spontaneous activity. Some neurons here fire very slowly. This is only the maximal amplitude of the response. Okay? And if you compare now the virgin group to the mother group, you can see that the data is plotted all over the place. So basically, this means that neurons change a lot in the mothers while they do not change in the virgins. Okay? If anybody has, now if you, as a speaker, sometimes you will prefer that people will ask you questions during the talk. Normally, Israeli students are, or the environment in Israel is such that our patience is low and we have to say what we think and want to understand immediately. Somewhat childish but true. But if you prefer that people don't ask you questions during your seminar, you can do that. Great. So basically, you can see just by even looking at the raw data, you can see that in mothers it's scattered all over the place, while in virgins it's on the line. What the authors next did is to calculate the distance of each point from the diagonal to quantify the differences. And when they, this is, excuse me, more examples. So if, I forgot your name. Matan was bothered by the heterogeneity in the examples. Heterogeneity is much worse in real life. And this is how the real data looks like. This is 6 different neurons. You see this is a neuron responding before, this is all in mothers, responding before and after odor. Before and after odor. So some neurons, have a very narrow receptive field and will change only in the intensity. Some of the neurons, there is a dramatic decrease in spontaneous firing rate and nothing much in the evoked. There is some in the evoked, but not much in the spontaneous, etc., etc. Okay, so real life looks like this. And if we quantify how far each neuron was from the diagonal, you can see that there is some noise in the virgins, which is shown here in the white bar. But in the mothers, there's always a significant change from the diagonal. If you saw, this is an absolute value because some neurons increased and some neurons decreased, and there is a whole lot of mess in the mothers. But in the virgins, what we considered here is a level of noise. And this is data from the spontaneous activity. And what the authors next did is did several controls. First is compare. So this bar is recording from the auditory cortex in virgins, comparing air versus pop odors. This is the level of change. This is in virgins comparing air to air, air to air. So this is basically the noise level, which you can see that there is some noise. If you go back to the diagonal, there is some noise, which is experimental noise. When you do an experiment, sometimes things change, or maybe even biological noise. But in any case, the author called this noise. And then they did two more controls. One is to ask whether pop odors is the dominant stimulus that causes the changes, or any other odor will cause that. So they tested two different odors. One is nesting material, which has a strong odor, and also other odors which they verified that are activating the olfactory system, an odor called acetophenone. And both of these did not affect the auditory cortex. This, of course, does not mean that the pop odors alone are the only ones who can affect it, but at least it means that not any odor that activates the brain causes changes all over the brain. And the other control was to look in mothers in a different part of the cortex. Okay, they looked in S1 in somatosensory cortex and saw that there's no changes in somatosensory cortex. This, again, does not mean that there are no changes in visual cortex, but it means at least that there is not a whole brain-wide phenomena. When the mother smells her pops, then something dramatic goes on all over the brain. And this is true also for the tone-evoked activity, and this is all statistically significant. The next experiment that they did is to try to separate behaviorally what is the component of the odor and what is the component of the sound. So they did a behavioral experiment where they washed the pups. So they stole the pups from the mother and basically dipped them in like a bathing thing. They washed them, and so basically the odors from the body of the pups, they go down. Just like after you take a shower, you have a good smell from the shower and not your own bodily odors. And there was a 50% decrease in the percent of retrieval of the mothers of the pups. So if you just clean the pups, so to speak, from the mother, there is an evident significant behavioral change. Interestingly, if you compare the time it takes the mother and to bring the pups back, this is shown in the gray dots and the blue dots for bringing back the washed pups. So even the mothers that did bring the mothers back home, it took them about 2.5 minutes, almost 3 minutes, until they were convinced to bring the pup back home, the first one. But then they rapidly, by the 5th pup, they already brought him back quite rapidly. And mothers with intact smell bring the pups very rapidly, like you saw in the movie. Okay. The next thing that they did is added 2 experimental groups. One experimental group they call experienced virgin. This is basically the sister that was brought into the cage with the mother and spent 3 days with her. Okay, this animal was never pregnant, never gave birth to the pups, never saw a male. She was only 3 days together with the mother. The other group was what they call mothers following weaning, and that is 1 month after the pups grew and went on to college, and the hormonal state of the animal is now back to basal levels. And again, this is the bioassay. You can see very clearly that both these experienced virgins and the mothers following weaning retrieved the pups very reliably. If you saw is that a similar thing that we saw with the wash pups happens here, is that it takes these 2 other animals more time to retrieve the pups. So mothers are great. They're the fastest. And now let's see if we can see the same changes electrophysiologically that we saw in the mothers in those two other groups? And the answer is yes. Yes, shoot.
I noticed that some of the graphs weren't the exact ones from the paper.
Exactly, yes.
So should one be able to add new graphs?
Yes, okay. So it's not graphs that I made up. What I did is I cut and paste graphs from the paper, okay? So for example, this is from the supplementary information, because I thought it's educational to show the students some real-life data. So I took this from there's a huge panel in the supplemental. This is part of the panel from the supplemental. This part was also only partly shown in the paper as, figure, I don't know, 4E or something, I don't remember, on the bottom, okay? And I didn't think that it's worthwhile to exhaust everyone with all the details. I just want to convey the points, okay? These are the controls of the experiment. So I put controls. I took from the paper one panel and put it as a standalone. Because normally if you take a paper and you throw all the figures on the board, it's very confusing. So it's hard to handle all of them. Especially the papers that you're going to deal with are going to be from the fancy papers, from the journals, from Nature, Science, etc. Normally they have, from A to J sub-figures, and it's extremely difficult to read the data. Now for you, this is rather easy. Everybody can see the fonts, etc. Which brings me to another issue, is fonts and colors. Minimum, never, ever, ever use less than 16 Arial. Okay, the font size, never. It's not readable by the audience. If you have something on the board which is less than 16, it will only interfere and you cannot read it. The other thing is colors. Try to use colors which have a good contrast on the board. Don't use red on black or red on blue, etc. Psychological tests show very clearly that black and yellow are the best combination in terms of contrast. But I'm a little bit sick of black and yellow, so I put this in white. Okay.
Put part of the graph and then show the rest too, right?
Yeah. You can do whatever you want, no problem.
You just cut it.
You can cut, just crop it, cut and paste. Or another trick is to put a white thing here and use the animation as an exit. So you put a white rectangle, and then you just disappear, and something new is shown, like I'm doing here. Okay, so basically this shows you data from mothers and virgins, which is basically what you saw before. But now they used a new protocol, which has to do with natural sounds. So they played ultrasonic vocalizations, wriggling call, and broadband noise. And you see an example from the ultrasonic vocalization, the real raster here. And this is before and after pop odor presentation. You can see the differences. And in this case, the differences come about in the threshold of the response. You can see that each stimulus was given 3 times at 3 different intensities, high, medium, and low. And you see that, for example, here, when you give the animal a wriggling call before, there is this response which goes down. And with the pop odor, the threshold simply moves, and now the responses are stronger to all these tones. Okay, so this is just an example of 3 different stimuli in mothers and in virgins, and the data holds as before. Again, this is raw data. Now only to 3 different stimuli, not the whole 1 to 40 kilohertz. But the argument is that some of these are natural stimuli and may be interesting. If you look at the other groups, same thing holds. So we see physiological differences in the experienced virgins and in the mothers following weaning. And again, this is how real data looks like in the brain. If we look at the statistics hold. So the naive virgins, there is some level of noise. This is, again, this modulation index from the spiking activity. And you see, you saw the data before of the mothers. You saw this versus the naives versus the mothers. But the different groups also show this statistical differences. One thing to notice is not only behaviorally, but also electrophysiologically, there is a significant differences between the mothers and the other groups. Okay, so we saw that in the retrieval time, and we also see it here in the electrophysiology. Okay, I think because I am giving the narrative of this talk, I'm a little bit short on time, so maybe I'll use this as the last slide. I think I have maybe 3 more slides or something. So what they tested here is these 3 different calls. One, just changes to broadband noise, wriggling calls, and USVs. What you saw before is that changes were all over the place. This is why we use this absolute modulation index, okay? Because some neurons went up, some neurons went down, and now they wanted to test whether for specific calls, let's say the calls of the pups, there is a homogeneous change. And in fact, what they saw is that, again, in mothers, this experiment was done only in mothers. Changes are all over the place for just broadband noise and even wriggling calls. But for the ultrasonic vocalization, the points fall on the left side of the diagonal. There's a shift. So basically, for ultrasonic vocalizations, there's largely increases in responsiveness, and this is quantified here by the absolute modulation index, by the non-absolute, by the real modulation index, and you can see it averages out in the different calls, but it pops out as significant in the ultrasonic vocalizations. And if you want to look at raw data of the sounds, in fact, it is quite difficult to find neurons in the cortex that respond to ultrasonic vocalization. In fact, they found only 15 of these neurons. All of the neurons are shown here, and they're shown before and after the raster plots, before and after the pop odor was presented. And the red arrow shows increases in response, statistically significant increases, and the blue shows statistically significant decreases, and some neurons did not change, very few. In fact, only 1, and most of the arrows you see are red, meaning that the changes were as increases in firing rates. I'll skip the next part, which is the 2 slides, and then you give a summary to the class. What have we talked about today? We talked about 4 different points. 1, natural sounds and smells interact in the primary auditory cortex. This phenomena is found naturally in mothers. The interaction can be evident after experience with the pups. You don't actually need to be pregnant and give birth to the babies, but it's the learning experience or the experience with the pups that can trigger this. The last thing was that changes are particularly sensitive to these behaviorally relevant stimuli. It's not just that 11 kilohertz tone, which I showed on figure number 1, are going up. These go up and down, and something happens in the cortex. But if you look at the real calls of the babies, there are more homogeneous changes. And also there is a subtype of neurons that are changing, which I did not have time, which is the last slide. And then you go, if you personally have problems with the papers, if you have open issues, if you have any comments, if you have a follow-up, and we are expecting you to give your own opinion about the paper, okay? It's very rare that students of the ICNC program think that, or any Israeli for that matter, that the paper was good or convincing to some extent. So you have to dig in and find what is bad about the paper, where are the problems you think, what other follow-ups. And I'm sure we can do that on this paper, but we will not do that. We'll take a break instead. But before, the last thing I want to say, which I didn't mention, is to keep the number of slides. The maximum is 1 slide per minute. And we're going to talk 40 minutes or 45 minutes. That's what you're going to get, or actually 30 minutes. So what I gave you now is a 40-minute talk, 45-minute talk. You see there are 21 slides here, 22 slides. This sums up to half a minute, so 2 slides per 1 minute, okay? You can go half a slide per 1 minute, okay, whatever. You could never ever have more than 30 slides. For your talks, you should have 15 slides, okay? And this means that you're going to have to work hard at home to, scrape off material from the papers that you read. Don't give us everything. Give us what you think is important. Give us some of the real data that that the class will be able to appreciate what has been done and whether the conclusions can support the claims of the paper, or the experiments can support the claims of the paper, etc. This is the most important thing. You have to, you are tested on on the presentation itself, how well you understood the paper, how well you can answer the questions, how critical you are, how elegant and aesthetic did you convey the message. And there's a very simple test. Did the person sitting in front of you learn something new today? Because in many cases, we sit down, think about 50% in talks and meetings, etc. 50% of the talks blank, as if I was not there. I don't understand what the person is talking to me at all. Learned nothing, zero. This is a bad thing to happen. Okay, you don't want to show that you're smart and you can do mathematics or whatever. This is not good. You have one goal, and the goal is to convey the message to the people. If it's a complicated issue, it involves mathematics, you have to solve the thing on the board, do it. But note that if you're dwelling on a slide for a little bit more time, you're going to have to spend a little bit more time on it. And that's fair, and that's not a problem. You can spend, 5 minutes on one slide or on 2 slides. That's not important. It's even actually better if you can convey a complicated message to the class. But don't put everything. And remember, less is more. So you have to scrape off from the papers the more relevant points and bring those up for discussion. Any questions? Okay, I think I said everything I wanted, and I put this thing on the board, on the website, and we're going to take a 10-minute break, and then we're going to discuss the paper of Yubel and Wiesel.
While running late, do you suggest run through what you have left, or to skip?
Okay, if you're running late, like I was just running late, then it's better to skip, fast a few slides and just, disregard one topic. But don't do many topics very fast. Don't say, and this also, and this also, and this also, and this also. Okay? So just, say this, I'm not going to talk about at this point, and go faster. לא, היה לי עוד סטודנט כמוך שבא אליי לפני השיעור.
קראתי את האלה של הפעם הבאה. אתה יכול לעשות את זה שקף 2 מהפעם
לא
הבאה.
הבנתי, היית צריך לקרוא את המאמר שלהם, גם לא קראת.
כתבתי את המאמרים של הפעם הבאה, אתה יכול לשאול
לא
עליהם.
הבנתי את
של אילן סגל.
למה?
בטעות.
אוקיי. כן.
בקשר לשאלה שלי לגבי המאמרים שבתלונות שנחתכו לפני הזיווג שלהם, לבין אחר כך שהתייחסת לאקספרימנט האלה, אז האם זה נמדד גם אקספרימנט ורזן ביחס לאימהות, אבל לגורים שהם לא של האימהות?
אני מבין, אבל לא. 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 זה נמדד רק עם אותו סטריין, עם האחיות שלהם ועם ההורים שלהם.
Hi, I'm a person here. This is how it's actually for you.
So you're part of this course?
No, so it's only physical physiology, so this is like the
Okay, what's your name?
the last name, yeah. Take it.
Yeah, I will take it.
Thank you.
Yeah.
היה אתמול שאלות? היה עדיין לא
כן, היה
קטע
תרגיל
של
תרגול של הרשתות.
Yeah.
I always have a problem with presentations with the images sometimes that you borrow from the internet. They could be sometimes copyrighted and things like that. How do you deal
אוקיי,
with
זה טובה. זה טובה.
So I think in academia it's not a big
כל
deal.
האימהות.
Yeah, nobody will give you any trouble. But if you, but sometimes I put the source, okay, or the website, just images, on the web or something like that. That's not a problem. But sometimes you just put the source on it. But if you use it for academic purposes, it's normally not a big deal. Nobody gives you problem with that. Nobody is going to come to arrest you because you showed an image of a neuron from X or Y. הלכתי לקרוא את המאמר. חשבתי, הייתי ילד טוב, קראתי אותו. של יובל וויזל, את שלי לא קראתי, רק כתבתי. אוקיי. Attention, מיקי יש לו מה לומר.
Everybody's waiting. Tel Aviv. Point for Aviv.
Are you waiting for me?
Yeah. And okay, I just wanted to add like 3 points, minor points, but are very important, I think, for the things that you're going to do. First of all, and there might be obvious for you, but just in case, if you have a paper to present, it basically means that you usually need to read more than one paper, because like the background could mean that you need to read like at least 3 others, or a review paper or something, because these papers from Nature and Science usually have like a lot of background behind them that you need to somehow have a very good grasp on if you want to present it to an audience that never heard of them before. That's one thing. The other thing that is extremely important in a presentation like that is rehearsing. Even if you have 20 years of experience behind you, you have to rehearse the talk. It doesn't mean that you have to remember everything by heart, but the amount of that you say in a talk, if you haven't presented it before, until you see yourself on a video, you can't appreciate it. Because like all your consciousness is gone when you're presenting something in front of an audience, and you don't hear yourself. So you have to do it many times before. Different people do it different ways. Some people rehearse the whole thing from beginning to end, and this is why they don't like to be interrupted, because if they're interrupted, they have to start again from the beginning. I personally cannot actually do that. I tried many times going to do like a mock talk from beginning to end. It's very difficult. But what I do is I rehearse every slide, which I think is at least it helps you during the slide to say it again and again and see that, with 3-4 repetitions, you see that you make it much better. And the last thing is that you shouldn't only use 16 and up font. You should never use Comic Sans font. People do that, and it's horrible. Really. So that's a really minor, but it's really important.
Yes, Ariel is the best.
Ariel, Helvetica.
Helvetica is okay. So, basically, the design of the course is that you're going to give one talk, which will be 10 points of your final grade, the one we just discussed. The variance is not too big. I mean, it's between 92 and 100 normally, because normally the people are good and they come prepared. And the other 10% of the grade should be the discussions of papers. Now, from the number of people in the course enlisted, I don't think we're going to have too many of these. Unfortunately, we're not going to have too much time for too many of these. But this is the first time I ever do this. I don't know exactly how Eddie did it last year. It should not be unpleasant experience. It should be a pleasant experience. However, we should do it in a discussion way, panel, rather than me asking questions or leading the discussion as a whole. And let's make a deal that if you did not read the paper that you should in time, not today. Tell me, and that's going to be easy. I'm simply going to decrease that from your 10-point grade or something in some way, and so you don't feel uncomfortable. And again, it's very hard to grade this. So I again assume that the variance in grade is not going to be dramatic. So it's going to be a few, if you know what you're talking about. Because the idea is to learn and discuss these things together and to share ideas rather than to give you an exam. However, experience proves that if I'm the only person who read the paper and the discussion is not very high level. So this is why this is our mean way of forcing everyone to read the paper. Okay, so today we're going to talk about a classic paper by David Hubel and Thorsten Wiesel, one of the classics that got him the Nobel Prize. I think that this paper was cited over 7,000 times, something like that, which is unbelievable. Okay, if we have a paper which is cited over 20 times, we are very happy. It's considered a big success, and this is really, really a classic. Huh? So, what is the paper about? Benjamin. Who is Benjamin? Benjamin. Shoot. What do you think? What are we talking about today?
We're talking about the electrophysiological responses of neurons in the cat's visual cortex to both simple and complex visual stimuli.
Okay.
And what is the organization, and what is the, the detailed properties of the neurons in the visual cortex.
So there's some specific hypothesis that they put up front to test, or this is largely exploratory?
Well, I felt like it was more exploratory than anything, but I think their hypothesis is whether the lower relay station in the visual system, whether it directly conveys information to the first processing station in the neocortex.
Okay, my feeling from reading it is that it is very exploratory, they're very creative, and they have a deep feeling for each cell that they're recording, and they are just making their way through into the forest, and they find this and they find that, and they try to categorize it. Very, very clear that it's very unquantitative. Okay? Almost no statistics, very poor on that, and I think it's very exploratory. So you like the paper?
Yes, I think that they're what's the
They
word?
could have minimized it too.
Yeah. Also, it's very long. These days, you are no longer
Right.
allowed to write so much, your thoughts and things like that. So it's true. It's a little bit longer. It's longer than your normal paper. Although if you read our neuron paper, which is obviously more recent, with the new rules of the journal, it's also, it's better. It is not big. But yeah, it's definitely quite long.
What's amazing is that they describe it as a hunch, as things that they feel, and they were almost always correct.
Yeah.
But that could be revisionist, though. If they want to make themselves look better, then they thought, look at this, we predicted it would look like this.
No, but
Okay.
like you said, there's no big statistics, but still they get to understand of anatomy that we know today that is true. So,
Okay. So we're talking about the striate cortex. Striate cortex, you know what striate cortex means? Okay, it's striated and fuscous. And this is for V1, visual cortex 1, which is where in the visual pathway? Nikki, shoot. Where in the brain are we talking about? Did you read the paper? Who did not read the paper? So I won't bug him. So Leah, right?
I didn't read the last half.
Last half, first half. Okay, let's start with the first half. I doubt we will get to the last half. Maybe. So who else? Enrica, also
Yeah.
first, right?
But I know about this research. I didn't actually read this paper, but I know
Okay.
the story.
Okay, no worries. אז הרן, אמרנו, ובישארה גם כן. אוקיי. You too. Okay. So we'll skip you. So, you know, the paper is 3 parts. Okay, first part is about, let's see, Adi.
Hi.
ככה אני זוכר.
Yeah.
First part of the paper, what is it about?
I think it's they try to characterize the receptor fields, and they discovered that they termed it as simple versus complex.
Okay.
And
Okay, so that's great. So the first part of the paper, I think, is entitled Organization of Receptor Fields in the Visual Cortex, Properties of Simple and Complex Fields.
right.
Great. So, part 1, this is just the recordings where they stuck their electrodes. Also, extremely, before you go up to the board, you can come up. Can't come.
Okay.
So basically, just before we dive in, you see that they used 40 cats, 40, which is also something that would never be allowed today. The ethics committees are very strict, and you have to squeeze a lot of information from each animal before you kill it, especially if it's a cat or a monkey. And they used 45 electrodes, 45 penetrations to 40 cats. So just to take over 1 per animal. But basically,
Can I ask something about it?
yeah.
Why is there most of the electrodes only on one side?
On one side of the cortex?
Yeah.
Because they want to check, to be consistent. Normally, laterality in the brain is an interesting issue. Okay, in some aspects you have very clear laterality. So the right half of the brain is creativity and whatever, blah blah blah. But in many cases you don't. But in experiments you have to try to be consistent and do your recordings from normally the same thing exactly. Okay?
The few on the other side are just controlled
Yeah.
to make sure there is no.
Yeah, I don't remember. I read this paper yesterday. I don't remember if they refer to these, but
I'm asking because I didn't see it.
They did.
Paper.
they did not. Okay. So basically, it shouldn't matter in this case, but normally, experimentally, you want to restrict yourself, and also they try to be in the middle. Okay, so Adi,
Yeah.
tell us about what we see here in Figure 1.
Okay. So they show here.
If I'm too aggressive, you have to tell me. But
No, it's okay.
This is a trial. We're
It's
just
your target. No, it's okay. So there's they characterize it here to 6 types of simple and complex receptive fields.
No, these are only simple cells. These are
This is only the simple? Okay.
Only simple cells.
Not
Yes.
Okay, right. From the lateral genicle. Okay. So a receptive field. Should I need to explain?
You tell them, not me.
So the basic idea.
No, what are we
of,
seeing on the board? We see
Okay, so on A and B, we can see here that we have this is the concept of on center and off center. The X's are for A, for example, it's an on center when the light will be on this area.
This is
it
a
will
yarn
be
from the cortex?
activated.
A and B?
A and B are lateral geniculate and cortical receptive fields on center. Geniculate. From the geniculate receptive field.
So A and B are from the LGN.
Right.
Okay? And then
It's
C
on and off.
to G is from cortex. And first station in the visual system is the retina. Okay, and then you go up to
The thalamus, optic tracts.
the main next station, LGN, lateral geniculate nucleus, and the next station, right cortex or V1. Okay, but of course it's not only 2 synapses away, you have 6 synapses away. They refer to this in the paper because there are several synapses here. In the rods and cones we will learn about these, and these go to the thalamus, and these goes to V1. So A and B are receptor fields that you can find in lower brain regions, right?
Right,
Okay.
from the LGN.
Okay, and C and G are from V1. So tell us what do we see here first?
So, for example, in C, again, the axes are when the light hits on that spatial area, they are activated, and you can see the shapes that here it's like circles, but from C and D and also in E, you can see that they have this long rectangle slit, they call it, yeah, that they react to, and so.
Ahmad, that's good. Let's turn on. Let's get some more people involved.
Can I ask something?
Yeah, sure.
I was wondering, do the dashed lines indicate the...
Me, the matan? Okay, let's first get the question. You asking me or Adi?
No,
I
whatever.
don't know, whoever knows. I was just wondering if the dashed lines indicate the direction of the stimulus on the retina.
The rotation.
So what kind of stimuli did they use here? So matan, right? That's your name? Matan. תחליף את Adi. Okay, so what types of stimuli did they use to get these receptor fields?
They have lines, slits, and also spots, as I understand.
So here they made it with lines? Or
Uh,
slots or slits? I don't know. I think it's just
no,
spots on the visual field.
yes, because it has a very simple way.
So what's simple about these? So first of all, these are stationary. They start by just stationary, shining light on the screen in a stationary manner. And if you do this and record from the LGN, you see these on-center, off-center neurons. And if you do it in the cortex, you see this different configuration. And they call them simple. Why is it simple?
Well, you can see it as you have the areas of on and areas of off, like the circle is off and on in the LGN, but here it's not in the range of circles, but in the different range, and just which looks like this. And for example, if this is inhibitory, so if you have a spot here, this make the firing rate decrease, and if you have a spot here, the firing rate increases, and if you have in the same amount here in the inhibitory and excitatory, so they cancel each other,
Okay.
and give the result is like in the initial condition, like the firing rate of spontaneous firing rate, and you can also it also has the property of summing. So if you have spot here and spot here, so the in this case increasing, so the increasing is 2 times or something like this.
Okay.
2 times of one spot, and here for decreasing. From the same logic, you can conclude if you have a line like this, so spots here and here cancel the spots here and here. So
Okay.
you have the spontaneous.
So the simplicity of these cells means is that you have a very good predictive power on what type of response you're going to get based on these receptor fields. Okay? So they describe simple receptive fields, and they have different structures from the LGNs. They're now more elongated and have this main axis to the receptive field. And in many cases, you can see a concentration of excitatory centers, inhibitory surrounds. They can be of different sizes, different forms, and there is synergy, and there is summation, and there is predictive power. Okay, these are some of the statistics. We can skip that. Okay, and next they find something peculiar, which they call complex cells. And the first one is shown with, they show an example with stationary responses. So let's try to go to Noga. Who is Noga? Alan Noga. Okay, tell us about the experience of finding a complex cell. What's complex about this cell?
So let me first explain what you see here, and then I'll discuss the difference between this and the simple one. So they basically check the width of the stimulus and the location in the receptive field. So you see, the first one is the narrow line, and it causes.
Yeah, just talk to them, not to me. I'm all set.
And it causes an off response. As you can see, when the stimulus is a little bit lower, we see the off response is larger.
Where do we see that?
It's B.
So in B we have an off response.
In A and B we have an off response, and it's a narrow stimulus in the upper side of the receptive field.
Okay, so they flash this bar above in the upper part of the receptive field of the neuron, and in A and B we see a decrease. Okay, what happens in C?
In C we see a peculiar result, which is both on and off.
Okay.
Result.
So why is this bizarre? Why can't this explain what's complex about it? Because it can be, something like this. Let's say this would be tilted all the way through, right? And then you put a bar here, like Matan said. With this slit, you're going to get excitation, or off, 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 off. And if you put the slit around here, you're going to get this mix, off and off, or something like that.
No, because we said that the simple receptive fields are both, you can explain the reaction both according to summation, and you can predict exactly what the reaction would be. So I think if you put the line exactly in the middle, either the inhibitory and excitatory fields would cancel out.
So I have to write questions to your exam in the end of the semester. So if I give you A, B, and C alone, you can say if it's a simple cell or complex cell. That's going to be a question in the exam, not this particularly. But so also for the exam, we're going to not only the slides, also the papers that we read. We're not going to have what?
This paper also?
This paper also, yes. We're going to have like 3 papers for discussion throughout the semester, so don't worry. And last year I was in Stanford. Something on the average of 6 papers per week they had to read. I think only when they were in the shower the students didn't read. Candelisa Rice gave a course. Candelisa Rice, and there's a very, 400 people waiting list to get into the course, and they have to read like 20 papers per week or something. So they have no life. So 3 papers a semester is fine. We'll manage. Not that I'm Candelisa Rice. Okay, so let's say if you given the exam, what's your answer? Is this A, B, and C? Is it a simple cell or a complex?
I think according to C, it should be complex, because we had 4 coherent that defines the simple receptive field. So
Jonathan, what do you think? A, B, and C, it's a simple cell?
No.
It's a complex cell?
Yes.
Yeah, just based on C?
Yeah.
Because they don't cancel each other.
But just based on C?
No, if you have A, B, and C. I mean, if you have A and B, clearly, it can be a simple cell, right?
Well, don't simple cells, don't they have a specific location as well? So if it's moving, then you're
the edge example.
Does the like
Like FNG.
don't they cancel every distribution? So it can be that in one time off, one time on, one time nothing. You don't have any distribution. It's uniformly distributed.
Of course there is a distribution, but one of the criterions is that you can predict, I guess
Can
with
predict
some
that
noise.
the mean. Okay.
Right, but here you see
Can't it
an,
be the
an
noise?
on and then an off response. And if it would be
So
just
can it be a
summation.
noise deviation from the mean?
Well,
So,
we are giving an
Noga,
example.
when we looked at simple and complex cells, okay, did we ever see these kinds of diagrams for complex cells in the paper?
This is a simple.
This is a simple. Did we ever see these types of diagram in complex cells?
There are edge complex cells, but then the 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 border won't be.
No. Did we ever see in the paper these kinds of diagrams for complex cells?
Oh no.
No. Okay, right. So if I give you in the exam, is this a complex cell or a simple cell? You would say clearly it's a simple cell, right? Okay. Why? Because if you present now a bar on the side, okay, this is what I see here in A and B, right? We go down, we get a inhibition or off response. Inhibition, inhibition, inhibition. And now I put the bar here. And I start to get excitation, but also there's some kind of an off response.
But Adi, they say that an on-off response is the characterization of complex cells, and many complex cells had on and off response, and nearly no simple cells. Okay.
A few times,
Yeah.
yes. Okay, it's fine. Fine. No worries. Okay. But generally speaking, it would be a little challenging to say that this is complex cells up front. Okay,
But
one characteristic of
100%,
complex.
80% you can say it's a complex cell just based on the fact it has on and off response.
No, I don't think so that it says that in the paper.
Statistically.
But no, to have a complex cell, what does it mean to have a complex cell? That's my question. Not if it has a given pattern of activity. Because you can find in simple cells a given pattern of activity. If you can predict it, let's say if you have now these types of responses, and you put your bar or your spot of light a lot here and a little bit here. Okay? So normally you will get summation, these can
But
cancel out and it
if
will
it's a
make sense.
stationary stimulus, how would you explain the on
No,
and
I'm
the
not
off?
explaining cell. I'm not explaining that. I'm just saying that what is the definition, how they describe the complex cell? Why didn't they put these types of graphs on? A complex cell is a cell where you cannot explain, based on these, the patterns of responses. So for example, if you put this pattern of stimulation, you would expect to have an off response. And here you would expect to have a very strong on response. Okay, everybody is with me?
So F and G was it?
F and G would make them now very strong feeling that, or now I completely verified that this is a complex cell. Okay? This is also a first good indication, C. D and E, of course not, because you can fit it on some simple.
But you don't think that C is enough?
No. C is already a very good indication.
Adi, but how would you explain C? Just
No, I
for
cannot explain C.
information?
It's fine.
Okay,
Okay?
I'm saying that.
I'm saying that so one thing is that you don't have clear summation, which means, again, but maybe you can think of something, but here, clearly this falls, if you take C, F, and G together, this makes the complex cell a complex cell. Okay.
So in a simple cell, if you added D and E to get G, you would still have excitation.
If you add?
If it was a simple cell. So in G, there should be excitation in a simple
Yes,
cell.
a strong excitation.
Right.
Yes.
Okay.
Okay.
Summation of both.
So, okay, you can sit, right? And next, let's see another complex cell. Okay, one other thing which is very prominent in complex cells is that they love motion. Okay? Which was very clear, and you can get things like sustained firing in complex cells, which is something that you normally cannot get in simple cells. Okay? And what they do here is that they play with the stimulus, they go back and forth, and they get these complex cells, now with the moving slits. Before it was stationary responses. Okay, another thing is edge detectors that they find, and again that they cannot put an axis, a clear access to the receptor field. You simply cannot put a simple access to the receptor field. You can cross the axis and still get responses. Okay, what else is common or signifying a complex cell in general? Michal, Alan. You are now Torsten and sticking electrodes into the brain. Okay? What is in terms of the size of the receptor field? What would be common? Okay. So complex cells are, because of this, that their definition is loose also, that the receptor fields are larger. Okay, of these cells, which is very common. Okay, so now let's go, Michal, let's go on with you. And you can, so we have simple cells and complex cells. Complex cells largely is things that you cannot explain based on a simple receptor field. Yeah, okay, I prepared all of this, but we're not going to have time. Okay, that's the dimensions. Okay, let's go to part 2. Michal, what's happening in part 2 of the paper?
So we are checking the ocular abdominal. It's showing the stimulus to one eye or to the second or to both. What's the effect?
Okay, so what's the first station anatomically that we can find in binocular neurons? Okay, this is something you will see when we will talk about vision. You'll see monocular neurons, which are responding to one eye only, and neurons which are binocular neurons, which respond to both eyes. So most neurons that they find in visual cortex are monocular or binocular? Monocular? Binocular. Okay, they respond to both eyes, most of them. Okay, so now you have to explain. You can go up to the board and tell us what you see.
So,
and I outlined to your convenience the most important part, which is binocular synergy, and then you have to refer to its 3 cells because it's simultaneous.
Okay, so if you do the stimulus to one eye, and it's a moving stimulus, so you get some response, and if you do the stimulus to the other eye, you get the same amount of response, but if you do the stimulus to both eyes, you get a big response which is bigger than the sum, I think.
You think Nikhil understood what you explained? He didn't read it.
Okay. So
you have to be easy on us. We have now, we have 3 panels, A, B, and C.
Okay, so
what are we seeing in A?
So in A we have a stimulus only on the right eye. In B we have a stimulus only to the left eye, and we are checking the response when moving. The arrow shows moving the stimulus to what direction. Okay.
How many neurons are we recording here?
I guess 3.
Okay, that's a good. Where are how do you identify 3 different neurons on one trace?
Because I think they are different in the length of the spikes that they.
Okay. How come spikes are different lengths?
They show the cell number 1 with long lines, and cell number 2 with short lines, and cell number 3 just have a.
But each line is a spike, no?
Yes, each line.
So some neurons have. What's the size of the spike in a neuron? Normally.
No, it's the same size.
100.
So the membrane potential is what? Roughly. And the peak of the action potential?
40. 35.
Okay, it varies. Let's say the full spike is what? 80, 90, whatever. Something like that. Here the intercellular. Millivolt, yeah.
80 to 100.
80 to 100. So how come you have spikes of different sizes here?
Maybe it's outer with the background and the.
What? I can't.
In the third part, they describe background recording
okay.
and.
Now, what is the background recording they refer to in the third part?
Something else.
Okay, so these are actually neurons and actually units from single cells. Who knows how come you have different sizes of spikes?
Distance
Few
from the
records
electrode.
in the
What?
Distance from the electrode.
Okay, so this is not an intracellular recording. This is an extracellular recording. The quality of or the shape of the spike and the size of the spike will be determined based on the quality of the isolation between the tip of your electrode and the neuron. Let's say you're sticking your electrode, and this is not an intracellular recording. Let's say these are, now all of these things you have to, when you read this paper, you have to ask yourself, let's say this is a cell body, this is another cell body, whatever. And now you stick your electrode here. And you touch the membrane here, so you have very good isolation here. And you pick up the extracellular recordings from the second neuron. So one would think that the spikes from this cell would look very nice. The signal-to-noise ratio will be very high, and then the other ones will be low. And if you do statistics on your signals, you can verify that the shapes of these spikes are, and these will be individual in every recording, extracellular recording that you do, because they depend on the isolation between the tip of your electrode to the membrane of the cell. And therefore, in some of their recordings in the paper, they manage to record multiple neurons, sometimes 2 neurons, sometimes in this case 3 neurons, and you can see 3 different neurons. One with this size of a spike but is equal on both sides of the plus and minus ends of the electrode. This, roughly the same size, but you can see that it has different characteristics, okay? It has an upward deflection which is much larger than the downward reflection. I don't know if Dan will talk about, methods, I guess not. And these small baby spikes. Okay, so you have 3 types of response. Okay, so now let's see how the 3 different neurons respond.
This is the response of these 3 neurons to A showing the stimulus only on the left, B showing the stimulus only on the right, and in C the stimulus is in both eyes, and you can see that the reaction is much bigger for cells 1 and 2, and cell 3 only react to both the binocular, and also that you have a response, much bigger response to moving to the opposite direction too, here on that side.
Fantastic. So, Mishara, you understood her explanation? Roughly. Okay, so the idea is that there is synergy between the 2 eyes. So let's say if you look, first of all, what they found is that the responses of all the neurons, or many neurons, are binocular. They have the same receptive field, if they are, or the same properties, the same receptive field access, if they are stimulated by the right or the left eye. And what is the main difference between the type of responses, between one eye and the other? The strength of the response. Okay, your name again? Nina. Okay, great. So the most pronounced difference between the responses from the 2 different eyes is the intensity of the response, and not the properties of the response. Okay, what Michal showed you now is the property of the synergy, binocular synergy of the 2 eyes. How would you, okay, but this does not show the intensity, the different intensities of the response, so dramatically. On the contrary, you see, in this case, that neuron, the neuron responded to the left eye and the right eye in a similar manner. You see the number of spikes here and the number of spikes in one, roughly similar. But if you look on average on the responses of the different eyes, the most prominent phenomena is the intensity, and this is something that is called dominance, ocular dominance, okay? And this is, you will see this throughout the visual system. This is a concept that is called ocular dominance, and even there is ocular dominance columns, etc. And what they did, and they're going to use it later on, and we will hear it also in the video when I will teach you about the visual system, and also when I teach you about plasticity, is these types of categorization. Okay, so what do we see here? Let's see, Ada. Yes, Ada? Shoot, tell us what do we see here in ocular dominance.
They divided all the cells into 7 groups by
And the fourth column is the type of cells which respond equally to both eyes.
Like the one that we saw before?
No, the ones we saw before.
How would you categorize in this this neuron?
This one prefers it responds, which one? 1, 2, 3, which number?
Okay, I'm going to give this question to Nikhil after you explain what we see here.
Okay.
So listen. You're her exam. If you get the answer wrong, then she fails. If you get the answer right, she gets 100. Although today we're not giving any details. This is just the trial and error. Okay. So, Haki, your name again was? Adab. Okay, Nikhil. Open your ears.
So these are 7 different groups and of all the cells that they recorded. Cells in group 4 respond equally to stimuli presented to either eye. They don't care which eye. And the ones in group 1 prefer the contralateral eye, so they respond mostly to the contralateral eye, and almost not at all to the ipsilateral eye, and the other way around as group 7. And they also divided it into simple and complex, and you can see that the division of ocular dominance is the same in the complex type and in the simple type.
So do they have any quantification of this, of the 1 to 7?
This is the quantification. What do you mean?
No. So let's say, if me and you would have to score a given cell.
They said mostly.
Exactly. Okay, so this is another beautiful thing in this paper. So we see if it's exclusively contralateral, it's 1. Exclusively ipsilateral, 7. And now we're starting to enter into the gray area, okay? David thinks it's a 2, Torsten thinks it's a 3, whatever, and then they categorize it by, this would never pass an ICNC basic course without doing some serious statistics.
They even have a comment there that the division is not really, they say something about it, that the groups at the middle, they don't really know exactly.
Most of it has to confuse.
Okay. Leah, I know you didn't read it, but what's clear about this? What pops up from this graph?
What do you mean, like?
Now that you know what this graph is,
Yes.
what pops up?
The receptive fields of simple neurons is mostly binocular.
Binocular, mostly. I see a very clear skew to the left.
A very big what?
Skew. No?
Okay. If you say no, I think it's bigger, like when you say in a categorization of the 3 and 4. But
yeah. So most of the cells are either 3 and 4. But if you look at 1 to 3 versus 5 to 7, you see a very clear skew to the left.
Okay.
So this is another basic concept. These are the first people putting their electrodes into the visual cortex ever in an animal. Not ever. They also did it a few times earlier, and there's some things in frogs, etc., which unfortunately we don't have time to discuss. Frogs' visual system. And but you see a skew. So now the question to Nikhil: how would you put this neuron on the ocular dominance categorization? How would you score it? 1, 2, 3, 4, 5, 6, or 7? Neuron number 1. 1 is neuron number 1. The spikes which are.
Categorization, what do you mean? By simple or complex, or by
No, not simple.
Dominance.
Ocular dominance.
I think it's
I have to deal with this all my life, huh? At the end of the course, the students give me a score, and if they learn something new, it's good. So how would you categorize it?
I think it's dominant to the right.
It's dominant to the ipsilateral? To the right? that the responses of neuron number 1, which is this guy here, this is the responses to the left eye. This is the responses of the neuron to the right eye. You see that they are similar? Roughly similar. He gives, I don't know, 6, 7 spikes here, and 6, 7 spikes here. These people don't care at the moment for, if the temporal code, rate code, if the spikes are faster here first and then decay, etc. They just score, they just get an impression. And the impression is that this response and the upper response is similar to both eyes. This is one eye and the other eye. Okay? Great. So I would score it as number 4. Okay. So unfortunately, we cannot go on and on because I already owe you 5 minutes, I see. But I go on to do a third part, etc. This was just an exercise on how we're going to practice it. Did I not? I didn't ask everyone, huh? I have to get my list better. So excuse me. Next time I'll be better on this thing. But the idea is that basically read the paper. Let's discuss it as much as we can. And all the papers that we're going to discuss are going to, questions from them are going to appear in the exam. So it's going to be a very good exercise for us, for you to read them carefully, for us to discuss them. If there are any unclear things, they will get truthful, they will float up and we can discuss them. What are we looking at, etc. And hopefully we'll discuss some interesting and good papers. And this is like very basic knowledge for any neuroscientist. This paper on the functional organization. There are many. I actually wrote, 2 pages of questions to lead the discussions, etc. But we got stuck. So I think. So how do you think about this type of discussion? Stressful? Really? I was too aggressive to.
No, it's just stressful.
It was stressful.
The situation is stressful.
The situation is stressful. Okay, so you have to take relaxing pills before, do some yoga, or hike in the forest. I don't think it was too stressful. Obviously, they did not take Reshatot Bet yet. So I think it will be a good tutorial for your next semester. Yeah.
What about the papers you present? They are for the LSK,
No.
Only the one that we discussed?
Only the one that we will choose for discussion, and there are not, unfortunately, to be too many of these. And let's say, I'm going to give everyone 100 if there is some basic level of discussion. If you tell me, Adi, sorry, I didn't read, then we'll mark it down and that's it. But the expectation, I already see differences between people who read it thoroughly and not, people who can give me a fight when we argue about things on the board. And this is the level you should read it, and you should because this is something you want later on for your exam and for your own basic knowledge. So I think it's fair. I mean, the stress level, we can discuss it.
That's good.
I'm not stressed, yes. I'm not stressed, and that's good. And so we're going to start with Idan Segev, which is the non-stressful exemplar. Yeah, so we're going to start with Idan on Wednesday. He's going to give 3 seminars, and then, or something like that, and then you guys are jumping into the pool. And I'm still tweaking the schedule, so excuse me for all the changes. You might find yourself, in a different spot than you were before. Okay, have a nice day. כן.
האמת שגם קשה לדעת כאילו על איזה נושאים אתה רוצה להתמקד, כי יצא לי שהמתוד יהיה חשוב, ואני רואה שבכלל אתה תתן זה.
למה? גם וגם. הנה, דיברתי על המתוד, שדיברתי על האלקטרונים.
כן.
תשמע, זה מאמר כבד.
הפארט שלי, כאילו היה צריך להבין את המתוד לעומק בשביל להבין אותו, כאילו להסביר על הטיולים של האלקטרונים בתוך המוח, כאילו את המתוד היה גם כן בהתחלה.
אבל אם אתם צריכים לקרוא מאמר אחד, פעם בחודשיים, פשוט לקרוא אותו. קראתי את המאמר מאוד. אוקיי. זה לא יקרה.
למה?
למה? כי אני עושה את הקורס הזה פעם ראשונה, ואני אצטרך עוד להחליט עכשיו על מה אני אדבר בסוף הסמסטר ואיזה מאמר אני
אבל
אדבר.
כאילו יותר, אתה יודע, יש זמנים יותר נוחים, ואני פחות נוחים לקרוא מאמר, זה כאילו זמן שאתה
אני
שווה
אשתדל
שעות
להעלות את
רבות.
המאמר כמה שיותר. אין בעיה.
Do you think it would be helpful if maybe we change the seating in the room to be more?
I think so. I actually thought about that, and I think it's a good idea. Yes.
Also, do you think
So
maybe
it's like a circle or something like that.
if people can do it, maybe it would be better that we ask each other questions rather than you just being the one who's pointing out the ball.
I see.
I don't know. Just thinking.
Yes, that's possible as well. I mean, I can be a passive observer.
No, I mean you'd still ask, but you wouldn't be the sole person, pointing out and saying like.
So I don't
So
understand.
you
How
know
is
what I
that
mean?
going
Like
to work?
you would ask some
No, I
questions
don't
and
know.
other people would ask each other questions. More organic.
I don't understand how it is. I mean, I'm for it,
Like
but
if there's no,
I'm trying to
if people
understand.
don't have questions, you will have questions. But at the same time, when you have questions about if we go like if we do it like this way, where we go through the figures, I can ask, the person sitting across from me or something, a question about the figures.
Okay.
I'm just throwing an idea out.
Okay. I'll think about it.
Also, so the columnar structure, there's
Yeah.
still a debate ongoing debate, right?
Oh yeah. Columns, yes.
Yeah.
Yeah, this was part of what I wanted to talk about.
But in the recent paper is showing there is not columnar structure in the middle.
Yeah.
At least at a micro level, I think it is.
What?
I can't remember the debate.
So if you read, there's actually a few. So maybe if I give this brings me to the point that if I give a paper, maybe I'll also give some more modern reviews on debates or
Right.
things that refute this, or but then it becomes 2-3 papers to read, which may be good, but Alan. So I have to think about that. But anyway, if you look at things like Kevin Martin's recent papers and things like that, they say that there's no column, absolutely,
And that's with calcium energy?
no, with electrodes as well.
Apart from the barrel code.
No, but also, the barrel is very clearly, one-to-one with a whisker. So in that sense, you can easily identify a spot from the whisker pad onto the given barrel in S1. But also, retinotopically, you can put your electrode in the fovea region or outside with the retinal topic.
No, what I meant was apart from the barrel, I think there
No,
might
but
not be columnar.
even in the barrel, so it's true, the barrel is restricted to organization. But even in the barrel cortex, there are people who are very unhappy with the descriptions of columns or mini columns and things like that as a unit of computation. So there's a very good review, what is a column or something like that, even on the barrel cortex. I mean, it's clear that in layer 4 there are barrels. It's clear. Whether it goes throughout the column and this information, stays along the column, etc. This is debate. Now, the recent papers from calcium imaging, like ours, you see this on the board, shows that there is organization, but in the organization there's this salt and pepper organization. This is something I wanted to bring up in the discussion.
Thanks. See you in a month.
Yeah, I may come to Idan's talks, but I'll be back. Yeah, I think after February. For the
Okay.
factory.
We don't do Chakman.
Thanks a lot. Take it easy. Yeah. All right.
Go ahead.
Yeah.
Would you mind using the microphone in your place?
Using the microphone. Yeah? I don't mind. I think it works.
How are you doing?
Right. This is my first class. It's good.
Hello.
Okay.
Now you can talk really quiet and people will hear you.
Was I too quiet last time?
No, no, no.
Okay.
Just